Hello, we're glad you've joined us for this live webinar, Mission Possible, Automating NGS Sample Prep for Challenging Samples and Niche Applications. I'm Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Beckman Coulter Life Sciences. This webinar is the third and final presentation in a three-part series on laboratory automation solutions for smaller NGS labs. Those that are less familiar with automation who are interested in standardizing workflows and saving costs through improved efficiency and results. This year, Beckman Coulter Life Sciences is celebrating 30 years in automation as they continue to provide liquid handling solutions to help accelerate reliable results that drive scientific discovery. Beckman offers a full range of flexible and scalable automated workstations and integrated systems to automate genomic, cellular, protein, and other workflows in a wide variety of research areas, including agriculture, cancer and infectious diseases, cardiovascular, forensics, and neurodegenerative disorders. For genomic applications, Biomek-Automated workstations are complemented by a growing portfolio of nucleic acid extraction and purification kits built on the company's patented solid phase reversible immobilization, SPRI, magnetic bead technology. Let's get started. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. You can post questions to the speakers during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speakers, David Horvath, MS, and Zach Smith, MS, who are both Senior Application Scientists for Beckman Coulter Life Sciences. David automates proprietary and third-party next-generation sequencing chemistries. He has a Master's of Science in Medical and Molecular Genetics from the Indiana University School of Medicine. David's career experiences span a broad range of life science fields, including forensic DNA analysis, medical diagnostics, and agricultural biotech. Zach automates proprietary and third-party next-generation sequencing kits. He has a Master's in Environmental Science from Indiana University. Previously, Zach worked at the university's Center for Genomics and Bioinformatics as a project scientist. David will now begin his presentation.
Good morning, everybody, and thank you for attending this third part in our Small Labs webinar series. Today, as Judy mentioned, we're going to be talking about automating NGS sample prep for challenging sample types and niche applications. Just a quick recap of the first part of the webinar series. We reviewed our workflow for automating NGS applications and workflows. So this is our application development process. This webinar and the second webinar are available for replay online. And you can review the typical NGS workflow, what our general process is for scoping out, developing the applications, from market research to generating our data, application notes, posters, etc., and including the application installation and post-installation support. And we also talk a little bit about some of our new software features, including our Biomek method launcher and guided lab setup features, as well as DART, our data acquisition and reporting tool, which can be included as an enhancement to your Biomek software. The second part of our webinar series reviewed several DNA-seq and RNA-seq applications in the list here. And again, you can replay that webinar online as well. So today's presentation will discuss essentially 4 niche applications. We'll be talking about FFPE-derived RNA samples using automated TruSeq RNA access library prep, cell-free DNA whole genome sequencing using the Rubicon Genomics ThruPLEX Plasma-seq kit, excuse me, targeted Amplicon sequencing using Illumina's TruSeq custom Amplicon low input library prep kit, and then also HLA typing applications using Illumina's TruSight HLA sequencing reference panel. So to start, we'll discuss FFPE-RNA-seq. So RNA sequencing with FFPE-derived samples can be challenging. So fixed formalin paraffin-embedded human tissue samples are common in cancer research, and RNA derived from this tissue type suffers from a number of problems for NGS sample preparation. These can include degradation of the poly-A tail in mRNA species, which makes oligo-DT capture difficult, as well as degradation of ribosomal RNA species that may result in imperfect ribosomal RNA removal occurred using standard ribosomal depletion strategies. The chemistry that we automated was the Illumina TruSeq RNA Access Library Prep Kit, and the workflow for this kit aims at overcoming the challenges of FFPE that I just described by doing an exome enrichment versus the traditional poly-A capture or ribosomal depletion. So the exome enrichment is going to focus on the coding region of the RNA. Because of that, it requires less sample input. From fresh and frozen tissue, input requirements are only 10 nanograms, and for FFPE material, inputs are 20 nanograms. Because we're just focusing on the exome, the sequencing requires fewer reads, which can allow you to increase the amount of samples per sequencing run and also provide a more cost-effective transcriptome analysis. So to the right here is a graphic of the RNA access workflow. It's not intended on showing the detail necessarily, but just to show you the length of the protocol, the complexity of it. And the next slide kind of gives a condensed view of that workflow. So essentially there's the cDNA synthesis module. We have the traditional A-tailing and ligation steps. Prior to hybridization, we include a quantification, normalization, and pooling module. And then we carry into the exome capture, the hybridization steps, followed by a final capture, amplification, enrichment, and cleanup. So the challenge points, again, with FFPE material and with the RNA access kit, due to its length of time for processing and complexity of the preparation, is that we're dealing with highly complex and time-consuming protocols with 9 reaction mix additions, 6 Ampere XP cleanups, and 2 hybridization captures. Our automation solution eliminates the highly repetitive and time-consuming tasks with only 1.25 hours of hands-on time for the entire 3-day method. Low input sample requirements are challenging for this particular application, and the method supports either 10 nanograms of total RNA or 20 nanograms of FFPE-derived RNA with high recovery with our Ampere XP cleanup steps. So traditionally with Beckman applications, we provide an intuitive user interface to drive all the method options for our applications. So this application allows you to process any number of samples between 1 and 96. It includes 5 distinct modules with built-in start and stop points, which correspond to the Illumina recommended safe stopping points. We provide flexible index labware options and index transfer options. Index labware could include using the kits directly, the tubes directly from the kit, your own custom-array plate, and gives the option of doing automatic adapter transfers or a custom transfer by file. We include a quantitation setup module for picogreen quantitation following the first PCR amplification. That'll provide the steps needed to set up your picogreen reagent, and then once you get the results from your picogreen quantitation, it will set up the normalization and pool mapping for you before going into hybridization. Optionally, we have T-robot integration, which is the on-deck thermocycler, and that will increase the walk-away capability of the method in providing more time for technicians, robotic users to be performing more value-adding work versus tending to the automation to move sample plates on and off thermocyclers throughout the day. Along with our methods, we include a reagent calculator. So the reagent calculator will automatically provide master mix recipe preparation instructions to the user based on the number of samples and particular modules that were selected to run for that part of the application. It'll detail the labware configuration, the sample and reagent tube or reservoir fill volumes, and it updates automatically based on the sample number and method options selected by the user. So here I'll describe some of the QC data coming from the RNA Access Kit. So the images you're seeing here are bioanalyzer tracings following the first PCR enrichment prior to hybridization. So the upper left graph are overlaid UHR RNA control libraries following the first PCR enrichment. In the lower left, we have brain RNA control libraries following the first PCR enrichment. And in the upper right, we have FFPE RNA controls following first PCR enrichment. So as you can see, we have consistent sizing and relatively consistent yields from the replicates of each of those control types. In the lower right, we have the bioanalyzer tracings following hybridization. So this is going to be your sample upon completion of the entire RNA access method, just before getting ready to sequence. So we have 4 plex pool libraries, and there's 2 replicates for each of the control groups here following the second PCR enrichment. So again, consistent sizing and consistent sample yields from each of the control types. Sequencing metrics are as follows. We have a very high percentage of aligned reads. Greater than 90% of the captured targets were aligning to the expected coding region of the genome. On the contaminant reads graph on the upper right, the scale is 3.5% at the top. So what we're seeing here is the amount of contaminant or non-target reads. So we're looking at a very small percentage of ribosomal RNA remaining in our final library and reducing the amount of ribosomal component that's getting mapped and therefore increasing the amount of aligned reads to the coding region. Coverage, uniformity, and strandedness. We have very good, very high and consistent coverage uniformity across all sample replicates and control types, and also a high degree of strand specificity. Okay, on the alignment distribution, we have a high percent aligning to the coding region, so greater than 70%. A small percent aligned to the untranslated region, intronic regions, or intergenic regions. We have less than 3% abundance and less than 2% ribosomal RNA component, with an average of greater than 12 million pass filter reads on a high-seq run. The data is highly reproducible across sample types, including lower quality FFPE material. The kit provides low percent duplicates for the UHR and the brain control samples, less than 20%, and relatively low PCR duplicates considering the degraded nature of the FFP material. Highly reproducible median insert size is also seen across the replicates of all the controls. So in summary, the TruSeq RNA Access Automated method generates 96 highly enriched exome libraries in less than 3 days with only 1.5 hours of hands-on time. It includes feature-rich user interfaces and reagent calculators to create flexible workflows and reduce setup errors, and provides high-quality, robust RNA-seq data with greater than 12 million pass filter reads, minimal percent abundance, and percent ribosomal component, with highly reproducible results. The second challenging sample type or niche applications I want to discuss today relates to cell-free DNA. So what is cell-free DNA? It's also referred to as cfDNA. It's highly fragmented DNA in the blood that is shed from tumor cells, or can be shed from tumor cells. And there's a high abundance of cfDNA from tumor cells in cancer patients. So why is this important? So plasma, along with other liquid biopsies, are easily available sample types. And given that these sample types contain cfDNA, they can be used to interrogate cancer mutations or any translational research in oncology. In addition, research involving the fetal fraction of maternal plasma can be conducted using prenatal testing, non-invasive prenatal testing, and be able to interrogate the cfDNA in that sample material. So we've automated a cfDNA application using the ThruPLEX Plasma-seq chemistry from Rubicon Genomics. Rubicon chemistry generates NGS libraries from cell-free DNA isolated from plasma. It's optimized for low input cfDNA samples, less than 1 nanogram to 30 nanograms of input. It's fast and simple library prep process performed in a single tube, 3-step process. It provides extremely low PCR duplicates, providing maximum library complexity, and it preserves the GC representation of the input DNA. Cell-free DNA sequencing has several challenge points. With this particular chemistry, there's repetitive pipetting of enzymes, indices, and challenges during sample cleanup are prone to errors and fatigue. So our application provides full automation solutions, eliminating the highly repetitive and time-consuming tasks. And when combined with an integrated thermocycler, it becomes a complete walk-away solution. The low input nature of cfDNA library prep requires assurance of maximum sample recovery. So we have highly optimized pipetting to deliver highly accurate and reproducible transfers of small reaction volumes, ensuring the most robust reaction chemistry. We've automated the Rubicon Genomics ThruPLEX Plasma-seq kit on the Biomek-FX. While we use the FX for this particular application, it is definitely scalable to an NX, whether it be an enhanced multi-selective tip, multi-channel head, or a SPAN8 pod, as well as being transferable down to our entry-level instrument, the Biomek-4000. So as you can see on the left, the Rubicon workflow is relatively simple. The workflow on the left is essentially a 3-step enzymatic reaction that's additive to a single plate. At the end of that, the 3 enzymatic reactions, there's a safe stop point, and then the method can be continued from that point with the final PCR cleanup. We provide a user interface which gives the user options to choose various parts of the method to run. So there's flexible workflow options to either perform the reaction chemistry or the cleanup together or separate. It includes labware transfer options. It includes trusted Ampere XP cleanup and also has options for T-robot integration. The Rubicon Genomics ThruPLEX Plasma-seq kit also was developed with our new guided labware setup feature. So if you're familiar with the existing instrument setup, it provides an image of the deck upon execution of the run, but it's a little bit limited in being able to resolve stacked labware or to provide additional detail about labware setup. So guided labware setup is able to break down placement of labware on the deck, labware by labware, and provide any step-by-step instructions to the user, including how much sample to fill, where to place it, how many wells should be filled based on the number of samples you select in the user interface, etc. In addition, stacked labware is now able to be resolved through a profile view. So any stacked labware on the deck is displayed on the far right in a profile view. So you can see each level, each stack of labware, and the order that it needs to be placed on the deck. When you drill down into a particular piece of labware, guided labware setup has the capability to present the any information specific to filling wells within that labware. So in this example, we're looking at the reagent block, and there's recipe master mix preparation instructions. They're highlighted in the red and green boxes. Those recipes are specific to the number of samples that the user selected in the user interface. And then it provides instruction on the reagent block itself as to which wells are to be specifically filled with those mixes. Also, we have our new Biomek method launcher feature that's compatible with the ThruPLEX Plasma-seq kit. And what this does is provide a real-time milestone or update view of where you're at in your run, and provides information regarding the method parameters you selected in your user interface, a real-time view of Biomek labware on the instrument deck, and also allows you to have resolution of any labware data sets during the run. That can include barcodes, volumes, sample IDs, index numbers, etc., and you can interrogate those values in the course of your run. And lastly, the Biomek method launcher also provides a web-enabled live run monitoring capability via smartphone, tablet, or desktop PC. So we'll talk a little bit about the data from the Rubicon ThruPLEX Plasma-seq kit. So what you're seeing on the left are bioanalyzer tracings across a wide range of inputs from 0.1 nanograms to 5 nanograms. So you can see that there's consistent yield and consistent sizing from each of those sample inputs. That's due to the method being able to modify the cycle parameters depending on your sample input so that the final libraries come out with consistent yield. On the right, we're showing average concentrations of Plasma-seq libraries for 1, 5, and 10 nanograms input, as well as an NTC sample. So you can see there's highly reproducible results for each of those sample inputs, and then across inputs, the yields are consistent as well. Sequencing data for the ThruPLEX Plasma-seq chemistry shows a highly complex library with greater than 50 million reads per library, as well as low percent PCR duplication for 1 nanogram, 5 nanograms, and 10 nanograms being below 5% PCR duplication. 0.1 nanograms has higher PCR duplication than the 1 or higher nanogram inputs, although it's still extremely low, less than 20%, for an input of only 0.1 nanograms. So in summary, the automated version of the Rubicon Genomics ThruPLEX Plasma-seq chemistry on the Biomek-FX provides 96 libraries generated in approximately 3.5 hours from low input cfDNA. It reduces errors from repetitive pipetting, provides true walkaway capability with T-robot thermocycling integration, and provides highly reproducible results with low PCR duplicates with input amounts as low as 0.1 nanograms. And now I'll pass it off to my colleague, Zach Smith, who will describe additional niche and challenging sample applications.
Thank you, David, and thank you all for hanging with us for the second half of the presentation. I'm going to talk about 2 applications. One is around Amplicon sequencing, and the other one is about HLA typing, which could be considered a variant of Amplicon sequencing. So really, what is Amplicon sequencing? Well, Amplicon sequencing is a way of doing targeted DNA sequencing. It uses PCR to amplify discrete regions of a genome for high coverage sequencing, usually for variant screening. And so, for example, you want to be able to zero in and find specific groups of SNPs in a limited number of genes. So you would use this for looking at tumor characterization, for example. And other projects, such as the 1,000 Genomes Project, is using this to characterize SNPs and other variants found in various ethnic groups around the world. So it has a number of applications just for people. For agriculture, they're absolutely becoming a good way of typing crops out. and for cattle and such. So it's expanding into a wide variety of applications. So the kit we wanted to discuss as a way of providing an Amplicon solution today was the Illumina TruSeq Custom Amplicon Low Input Library Prep Kit. The kit is quite rapid. It's not a very long workflow at all. It allows you to generate up to 1536 Amplicons for each sample. The panels themselves can be created using Illumina's Design Studio software. So you would design your panel, and Illumina would send you a panel of oligos. And then you would take that in conjunction with the kit, and then you would do your library preparation. So just some of the potential challenge points that we see with this one. So accommodating a wide number of sample inputs. So our method user interface adjusts for allow you to run from anywhere up to 1 to 96 samples, and adjust the protocol automatically in terms of master mixes and labware, for example. And the other thing is the ability to incorporate inline controls. So the Illumina kit provides with a couple of inline controls, a positive control using 2800M DNA that's provided in the kit, and a negative RS1 control. We have the option in the method to use those or not. We would strongly recommend using them because Illumina recommends using them. But we realize that some facilities might not be necessarily interested in working with humans, so we gave the option of putting that option in the method. So our automation solution for the TruSeq Custom Amplicon Low Input method was developed for the Biomek-FXP, as you can see here on the slide. It effectively is 2 modules. You do your Amplicon library generation, and then you do your post-enrichment PCR cleanup, normalization, and pooling. The method itself, we do have a template version for the NXP Span 8, but the data I'm going to show you was generated on the FXP here. Regardless of which platform you're going to be using this on, the user interface would remain the same. So the user interface is shown here. It's basically just a couple of options to allow you to include inline controls, to run the double-stranded protocol as listed out in the method, or just run it single-stranded. You would enter in your number of samples and whether or not you're going to use an on-deck thermocycler or not. And then depending on the options you select in the user interface, you get a few other options on how to deploy indices and whether you want the instrument to automatically assign indices to the libraries, or if you would like to do that with a file-driven index transfer. And then how much volume do you want to pool for sequencing. So like the Rubicon method, this method also takes advantage of our new Biomek Method Launcher software, specifically the guided labware setup. So as you can see here, we'll be able to go through the deck and be able to see what it is where you're putting which labware. What in the case of master mixes, how to create how much of each component you'll need to create a master mix, and where to locate it on the deck. So we've got an example of the 24 position blue tube rack here, and where you would put all the various master mixes on there, and what volumes you would need depending on the number of samples you've selected. So just real quickly, I wanted to go through some of the data we generated. So we ran 2 automation runs with 86 10 nanogram per mega human reference DNA technical replicates and 10 water negative controls per run. And we used the TruSeq Cancer Amplicon panel for our Amplicon panel for this kit. The tape station trace you're seeing in sort of the center of the slide there is just an overlay of our various technical replicates. And then the positive control is up on the top for the inline positive control, and then the RS1 negative control is on the bottom. You can see we've got signal in our positive control and no signal in our negative control, which makes us quite happy. We then ran these samples through, each sequencing run was done on a single MiSeq run, and generated approximately 25 million reads for each run. The graphs on the top of this slide show the read alignment for each run, so we've got quite high read alignment for each of the samples. As you can see, it's greater than 90% for the vast majority of our technical replicates. And then the 2 graphs that are below show the panel coverage by library. So we do have some variation in the panel coverage, but it's not. I mean, it's approximately the average coverage for each library group was approximately 2,500x coverage for the entire panel. So the percent uniformity in the coverage was approximately 96 or 0.96. This data was generated using the Illumina TruSeq Amplicon app on the base space. So we then also took the base space data, and I wanted to see how we were looking at as far as consistency on coverage on individual regions of interest. So breaking it down into the individual 226 regions that are covered by the TruSeq Cancer Amplicon panel, I averaged out all the coverage for each of the 86 technical replicates. And as you can see, between runs, we had quite good reproducibility as far as in terms of coverage across regions of interest. So our run-to-run comparisons were quite good for the TruSeq custom Amplicon low input kit. Okay, so for HLA typing could be considered a variant of Amplicon sequencing. So, but why are we interested in it? So the HLA systems is the human leukocyte antigen system. It plays a significant role in the body's ability to recognize and detect foreign and infectious agents as well as malfunctioning cells. It consists of 21 genes and their various alleles. That reside on chromosome 6. There's been over 12,000 alleles discovered as of January 2015. And research for HLA typing includes a number of applications, including for bone marrow and solid organ transplants. There are a number of autoimmune disorders that are linked to variations in HLA alleles, and as well as certain cancers. So it's a very active field of research. So the vast majority of labs that are still doing HLA typing are still doing it with Sanger sequencing, and you usually have multiple assays or tests per sample, more than 30, and the assays are tedious, and they often result in ambiguous HLA typing calls, and false positives are even more worrisome. So the question was, how could a company apply NGS technology to HLA typing? And so Illumina has recently released a new version of the TruSight HLA kit called HLA v2, which is quite an improvement over the v1 kit in terms of the complexity of the workflow. But as you can see, it's still a fairly complex workflow. The workflow is here on the right side of your screen in terms of just what the bench operations that you would require to do. The kit itself covers 11 HLA loci, covers 3 Class A or Class 1 loci, including HLA A, B, and C, and it covers 8 of the Class 2 loci, as you can see here on the screen, DRB1345, DQA1, DQB1, and DPB1. And it covers all those in a single assay, effectively per sample. So the genomic DNA sample, you will amplify out the 8 Amplicon pools effectively per genomic DNA sample, and then those will be after the initial long-range PCR to create the Amplicons. You'll perform a cleanup on those. The next step is you will do normalization and tagmentation of the Amplicons, and then after tagmentation, they're pooled back on a per sample basis, and then there's an indexing PCR step, and then a couple of cleanups before it's ready to go onto the sequencer. So quite an improvement over the V1 chemistry. Some challenge points for this kit. The workflow is fairly complex and requires a substantial amount of manual pipetting, as you saw from the previous diagram. Our automation solution uses a modular workflow design, and it reduces the user interaction and allows for easy deployment in multiple laboratory configurations. So because the method itself is modular, we can take it and we can split it into whatever configuration is required for the lab. If they have a pre-room and a post-room, we can break it up that way quite quickly. Processing multiple samples, we can process anywhere from 1 to 24 samples within 2 days, within the 2-day time frame provided by Illumina. That's how many samples the kit will support. And we are using Biomek Method Launcher software with guided labware setup to provide intuitive setup instructions for reagents and labware, because we realize that setup errors do exist in the real world. So So here's just a quick map of the automation solution. So we developed this method on the Biomek-NXP Span 8 system with the deck configuration that you see there. And effectively, we have 4 modules. The HLA amplification setup is listed in green, primarily so you can split it off into your pre-room. And then we have HLA Amplicon cleanup, normalization, and tagmentation, and post-tagmentation for the modules. So the black arrows indicate places where you will have to interact with the instrument in order to put additional labware on the deck, tips, plates, etc. And if you're running more than 12 samples, the red arrows would indicate an additional interaction that you would have to do. If you were to scale this method up onto the FX, obviously there would be fewer interactions that you would need. To scale it down to the Biomek 4000, potentially more interactions than this. So we thought the NXP would be a good cost-effective solution for a sub 100K solution for HLA typing. This method is compatible with Biomek Method Launcher, so you would just add it to your launcher, and you would have your technician launch the method through the launcher rather than launching it through the Biomek software if you wanted to go down that road. The user interface is extremely simple. There's a drop-down menu that allows you to select which module you want to run, and you enter the number of samples from 1 to 24. And then, depending on which module you're running, you will get some additional options will pop up in the user interface. We're utilizing guided labware setup on this method. One of the things we can do with guided labware setup is we can very accurately display what needs to be put where on the deck. So as you can see on the image on the right-hand side of the screen, that's all of the HLA amplification primers. And we have color-coded the wells based on the caps that correspond with the Illumina protocol, so that then we can go through and the user can simply take those tubes for the HLA amplification setup or from the I5, I7 indexing, take the caps off, put them in the wells that need to go in, and put them straight on the deck. So here's some data from a run we did. We performed a 6-sample run with IHWG DNA provided by Illumina for 6 different cell lines. The bioanalyzer traces you're seeing on the left with the false gel image are the amplicons after they've been after the long-range amplification has been performed and they've been cleaned up. Excuse me. And then the bioanalyzer trace on the top is the finished library pools after they've been completely processed and ready to go on the sequencer. So we performed a single sequencing run using a MiSeq V2 300 cycle microkit and generated 4.2 million pass filter reads, which we were able to identify 96.7% of them based on barcodes. You can see below we've got the percent reads identified graph breakdown for each of the 6 library pools we were sequencing, and then some mean quality scores for the run. So generated a large number of high-quality reads. And then we passed the FASTQ data off to Illumina. They use their TruSight HLA Assign software 2.0 to do the HLA calls. And so this is the software is provided by Illumina when you purchase you can purchase the software through Illumina for actually doing the HLA typing calls. We identified only 2 ambiguous DQB1 alleles in 1 sample out of the 108 alleles that were sequenced total. And we compared a list of 98 alleles associated with our 6 samples. Our results were 97% concordant with the IHWG calls. There are 3 discordant or no-call results that we believe can be contributed to incorrect HLA reference types. So the reference type that was assigned to the cell line by the IHWG group was not correct. And this is something that we see quite frequently when we're dealing with HLA data from NGS, is that we can, because we are sequencing at such a higher depth than what you can get for Sanger, that there are more and more and more variants that are being discovered. And I believe Illumina has just released a white paper on this subject. So that largely concludes our presentation for today. If you have any more interest in our NGS applications, we have a webpage, info.beckmantheholder.com/NGS for more information, where you can see all of our validated and demonstrated methods and what platforms we've developed them on. And thank you very much for your time and your attention. And with that, we'll take some questions.
Thank you, David and Zach, for your presentation. So a quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. David and Zach will answer as many questions as time permits. The first question is, what is involved in moving a next-generation sequencing method between platforms? For example, moving between a Biomek-FXP and a Biomek-4000.
Okay, I can answer that question. So obviously there's major differences between an FX and a Biomek-4000. An FX has 2 pods, so we accomplish parts of the protocol in different ways using the 2 pods. So the multi-channel pod will be used for bulk cleanups typically. Our span 8 pod is used for enzymatic delivery. So when we scale down to an NX, we're moving away from a dual pod system to a single pod system. So both of our pods are capable of processing the chemistry, although the nature by way of which that the transfers are handled are just inherently different using a multi-channel and a span 8 pod. Same with the Biomek-4000. It uses a series of tools, a little bit different than what you see with the span 8 pod. So to convert down to the smaller platforms, the method structure and the workflow generally stays the same, and it's just a matter of reworking the steps to fit the hardware, essentially.
Are there other NGS-based HLA typing assays, and are you able to automate them?
Yeah, I can take that one. Yes, there are other NGS-based HLA typing assays, including the Immucor/Sirona chemistry that I believe has already been automated on the Biomek-4000 by Sirona. There's also NGS Go and Amplicon as well. We are able to automate any and every one of them on our platforms. Our Biomek liquid handlers are chemistry agnostic, so we don't really mind whose chemistry whose chemistry we're automating. So yes, we can take those.
What kinds of panels does Illumina TruSeq custom Amplicon support?
Yeah, I can take that one as well. So the TruSeq custom Amplicon panels are generated on the designs using Illumina's Design Studio software. For the low income or for the low input kit, they generally recommend restricting it to 1536 Amplicons per sample for those panels. I know that the TruSeq custom Amplicon version 1.5 can support several, basically tens of thousands, depending on what you want to do with it. So it largely just depends on what coverage you need and what model system you're working with.
How does Biomek software handle regulatory compliance? And also, is there a way of capturing electronic signatures?
Yeah, I can take that one. Yeah, so with Biomek software, we have the Biomek Accounts of Permissions, or BCAP, software add-on that you can put on Biomek that allows it to apply with CFR Part 21 compliance. So basically, you can restrict access to certain methods based on the user. You can also require electronic signatures for every time the method is changed. So basically, it allows you to track changes to the method and lock them down effectively. So.
And RNA.
I'm sorry. Go ahead.
Oh no, please finish. I'm sorry. I thought you had finished. Please finish.
Oh, please go ahead.
Yes, Judy. I think you can go on, Judy. I'm sorry.
Yeah, sorry about that.
Okay. Can RNA access methods work with highly degraded FFPE samples?
Yes, the RNA access kit has recommended inputs for different DV200 quality readings. So the kit is designed to work with samples DV200 greater than or equal to 30. So the 20 nanogram input that I described was using relatively high quality FFP material with a DV200 value of approximately 70%. And the kit gives recommended guidelines of increasing the RNA input as the DV200 value decreases to, I believe, up to a 40 nanogram input for lower quality FFPE.
Is the Biomek-FX a walkaway instrument, or do the operators have to babysit it?
I guess either of us can answer that question. So, of course, it's a walkaway system. Automation is going to allow the user to start the run and then let the automation do the work for you. The walkaway capability really depends on the workflow. For example, some processes are going to be broken up into separate modules depending on how much labware is on the deck. So if there's a capacity issue and you can't include all the labware from the very beginning of the 3-day process to the end, then obviously there's going to be breakpoints. With the RNA access method, it's a 3-day workflow, so there's breakpoints associated with the safe stopping points from the Illumina protocol. So it's walk away from safe stop point to safe stop point, and sometimes across multiple safe stop points, depending on the length of each of those modules.
Yeah. The other thing to consider is you can automate any NGS workflow in any one of our liquid handlers. But obviously the Biomek-4000 has got a much smaller deck, and it may or may not have an integrated thermocycler, although it can have an integrated thermocycler. And so depending on the hardware configuration at a particular site, you may or may not need to have more interactions with the instrument just to load labware. I mean, if you've got an integrated thermocycler and you've got an off-deck labware storage like an automated labware feeder or a Cytomat unit, then the instrument could, you could build it into a completely walk-away solution. It largely just depends on what the configuration is going to look like.
And the workflow tends to vary from customer site to customer site. So we're able to configure the walk-away capability of these applications to fit the user's expected workflow or anticipated workflow. Some customers don't actually want to run samples for an entire day beginning to end. They prefer to run in more of a modular fashion. So we create those breakpoints when appropriate and work with each customer to develop a workflow that fits their needs.
Is the poster available to view or the entire presentation?
Yeah, the entire webinar will be available on demand with LabRoots following broadcast.
And I don't know if at the Rubicon summary slide there is a poster screenshot. I think that might be what the question is referring to.
Oh, okay.
So yeah, that can be made available, and we'll be able to provide a link where the webinar link is to that poster.
Yeah, we also have a variety of application notes that we've developed for all these methods. So if you just go to our Beckman NGS website, you should be able to download application notes for each of the applications that we've discussed today. If you want more detailed view of the method, or you actually want to dig into the data a little bit further.
What is the difference between validated and demonstrated method?
A validated method is, we don't use the term validated. Validated is going to be a term used by our, more of a diagnostic term. Customer will validate their method in-house to meet very specific performance expectations of the customer. Our qualification status from our applications are either demonstrated or qualified. Qualified is actually a term that Illumina uses. It's not specific to all vendors. Each vendor that we work with may have a particular metric or data spec that they're looking to achieve when we're developing our applications with them. And each vendor will qualify or give a thumbs up on our application when it meets those specifications. When we say a demonstrated method, what we've shown is that in-house we've demonstrated that it provides robust performance. We've generated data and are able to generate data on the automated system that's comparable to a manual prep. In addition to just generating bioanalyzer data or quantification data, we carry our samples through the sequencing or the appropriate sequencing platform to show the robustness of the actual sequencing data and generate sequencing metrics around that as well. So kind of close that gap on what your final results are going to look like and how value-adding your data will be using our applications.
Is there an automated way to do quantification and QC on the Biomek?
Yeah, I can answer that. Yes, there is an automated way to do quantification and QC on the Biomek. We have automated a number of different ways of doing that. So we have automated, for example, the setup of the Agilent tape station on the Biomek. We have automated the use of fluorometric assays such as picogreen for plate-based assays. We've also automated the Kappa Illumina DNA quantification kit for quantifying the final libraries also on the FX. And that speaks to one of the things that I like about our liquid handlers is that it's not a purpose-built box. And so all the other ancillary methods that you need to do in order to get a sample ready for NGS sample prep, including quantification, arraying out of various sample tubes. If you're a core facility, you're probably getting lots of different tubes from various outfits, and they may or may not be labeled properly, but that's not something we can help you out with. But it performs all of those operations, and so you're not getting effectively a one-trick pony with our systems.
And so that's where you work with your field application scientists to really share your workflow with them beyond just the library prep need. So there's work upstream of that. We have extraction protocols that flow into rearraying protocols, which flow into quant protocols, NGS prep, the actual sequencing preparation, qPCR, all those particular processing needs can be accomplished with one system with multiple applications serving that entire workflow process.
Yeah.
Does Beckman Coulter have chemistries for nucleic acid extraction?
Yes, we do. We have a variety of chemistries that we sell for nucleic acid extraction, including DNA advance for isolating DNA from tissue. We have our RNA advanced chemistries for a variety of tissue types, including blood and tissue as well. We also have a kit called Formapure that we use for extracting nucleic acids from FFPE materials. And so we have a variety of chemistries that are built around our SPRY technology that we can use for isolating nucleic acids as a part of getting the sample ready for your NGS sample prep. So we're not just an automation vendor, we're also a reagent vendor as well.
What support is available for applications that are challenging to implement?
What support for implementation? So our demonstrated methods are typically built in collaboration with the kit vendor. In addition, we generally have customer collaborators as well to get the real-world story. What does data look like from real-world samples? So our FAS field application scientists will take those demonstrated methods that we develop with the chemistry vendor, with our collaborators, and then they work with the individual customer sites to configure to their specific needs, and we'll triage with the kit vendors and provide a holistic approach to support. So not just implementing the method, transferring on a USB stick to your computer, but making sure that that method is up and running, configured, pipetting optimized, and that we stick with you until you see that the data is up to your specifications.
Are all methods built with... Oh, sorry. Are all methods...
Go ahead,
Sorry about that. Are all methods built with your Biomek method launcher? And as a customer, how can I get a copy and have existing methods converted?
I can take that one. So we did use Biomek Method Launcher at SLAS earlier this year, and since then we've made it a standard part of all of our in-house developed methods. The launcher software is two components. You have the launcher which you interact with the method with, and then there's the guided labware setup functionality that shows you the image of the deck and the stack labware and so on and such. Guided labware setup, we can get that out to you fairly quickly. For the launcher, there is a fee for that one, but it's not very expensive at all. If you want to know more information about the pricing structure, just please give us an email, and we'll be happy to get you in touch with a salesperson.
Can a thermal cycler be integrated on a Biomek 4000?
Absolutely. Integrated thermocyclers can be included on all of our platforms, so the Biomek 4000, Biomek NX, and Biomek FX, with direct integration to the deck without requiring external shuttles, arms, etc.
I would like to once again thank David Horvath and Zach Smith for their presentations. Do you have any final comments?
Just thank you all very much for your time, particularly those of you who soldiered on through 3 of these with us. It's been fun, and hopefully we'll see you soon.
I'd like to thank the audience for joining us today and for their interesting questions. And we would also like to thank our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. The preceding 2 webinars in this series are available on demand on the LabRoots website. Part 1 is Easy Automation Solutions, Better Results, and Part 2 is DNA and RNA Sequencing Sample Prep, Automating Simple to Complex Methods. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through January 28, 2017. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.